Hello everyone, let's try to solve this question together. So basically non-invasive prenatal screening, NIPS is the abbreviation, that evaluates the genetic material, the free cell DNA. And what is free cell DNA? It is released by cells from placenta and is fetal origin, and this circulates in the maternal blood during pregnancy. So basically NIPS is basically checking the free cell DNA. So anyway, the NIP result is shown below. Pause the slide, zoom the picture. So based on the result, what is the true statement? There are 4 statements given. Read it through. What do you guys think is the right answer? Okay, the correct choice here is that an option of amniocentesis should be offered to the mother. So now let's see what was the result. So see result, basically, as you see that this test, which is done by this company, Myred prequel prenatal screen with amplified technology, and basically they check for or aneuploidy. So chromosome 13, 18, 21, and other like monosomy. So here they saw that it was positive, and the positive predictive value is about 54%. So let's see why the others option is wrong. So a detailed physical examination is needed. Of course, an examination is needed, but Turner syndrome, even with the phenotypical feature, there has to be a confirmatory test, either in the form of karyotype when the baby is born or amniocentesis before the baby is born. So examination has its own role, but genetic testing has to be done. So that's why A is wrong. A high positive predictive percentage precludes the need for confirmatory test. No, a confirmatory test has to be done because these are just screening tests, so confirmatory test has to be done. And of course C is right. What about the result is most likely false positive, so no further action. There is, of course, with this percentage of positive predictive value of 54%, there is chances that this could be false positive. But the second component, no further action is needed, is wrong. Once you get that, you have to offer mother amniocentesis, or at least after birth, follow the baby clinically, and also perform genetic consultation. So a brief note about monosomy, that this is of course a monosomy X. This is Turner syndrome, as you guys know, and you guys know the features of Turner syndrome, that is associated with coarctation of aorta, and there are certain features. So what free cell DNA, we talked about it. This is the fetal cell that has been broken down from the placenta, and that circulates in the maternal blood. So, and here it says that the positive free cell DNA result is not diagnostic. So see, because this is a screening test, and this goes with any screening test, that screening tests are not diagnostic. So, and again, this talk about the positive predictive value, that between 10 to 14 is important, and here we have 54. So that's why you have to do. And again, it says here that amniocentesis has to be offered to all mother with positive free cell DNA NIPS. Okay, so I hope that you guys will look at this reference. Here is this a nice video which explains how this process is done. And please do like the video, and if you like it, please do subscribe to our channel, and you guys have a wonderful day. Bye.